Have you ever wondered if it's possible to grow a brain in a lab? Let's embark on a fascinating journey into the world of brain organoids. Picture this: tiny self-organized three-dimensional tissue cultures derived from human stem cells, growing and developing much like our own brains. These are brain organoids, a marvel of modern science. They're not quite brains, but they're not just clumps of cells either. They're somewhere in between, offering us a glimpse into the intricate workings of the human mind. They serve as a revolutionary tool for studying brain development and diseases, testing drugs, and perhaps even unlocking the mysteries of consciousness itself. The potential of this innovative technology is vast and wide-ranging, from treating neurodegenerative diseases to repairing brain damage. This is not science fiction, but the reality of brain organoid technology. To appreciate the marvel of brain organoids, we must journey back to their origins. Picture, if you will, the early 2000s. The human genome had been mapped, but our understanding of the brain was still largely a mystery. It was a frontier waiting to be explored, and a group of intrepid scientists were about to embark on a journey that would reshape our understanding of the human brain. The first successful creation of brain organoids happened in the year 2013, a significant leap in the field of neuroscience. A team of scientists led by Dr. Madeleine Lancaster at the Institute of Molecular Biotechnology in Austria managed to generate three-dimensional cerebral organoids from human pluripotent stem cells. These organoids, while not perfect replicas, mimic the early stages of human brain development. This was no small feat. The process required a delicate balance of conditions, a precise cocktail of nutrients and growth factors, and a whole lot of patience. But the result was nothing short of revolutionary. For the first time, scientists had a tangible, three-dimensional model to study the intricacies of the human brain. The contributions of scientists in this field cannot be overstated. The likes of Dr. Lancaster, Dr. Yoshiki Sasai of Japan, and numerous others have been instrumental in pioneering this technology. They've not only opened the door to understanding diseases like Alzheimer's and autism, but they've also sparked a conversation about the ethical implications of creating mini-brains in a dish. But let's not get ahead of ourselves. The early stages of brain organoid technology were filled with trials and tribulations. Scientists had to overcome numerous hurdles, from figuring out how to keep the organoids alive to understanding their limitations. It was a process of trial and error, of perseverance and determination. This marked the beginning of a new era in neuroscience and biomedical research. The dawn of brain organoids had arrived, and with it, a new world of possibilities. From their inception, brain organoids have come a long way. As the nascent seeds of organoid technology were planted, these miniature, three-dimensional cellular models were rudimentary and limited in their capabilities. However, over the years, as our understanding of neuroscience and stem cell biology expanded, so too did the complexity and functionality of organoids. In the early days, organoids were simple spheroids, capable of mimicking only the most fundamental aspects of organ function. But as the years rolled on, scientists began to unravel the intricate dance of cellular differentiation and growth, leading to organoids that more closely resembled their full-sized counterparts. By the 2010s, breakthroughs in bioengineering allowed for the development of organoids with multiple cell types, mimicking the diversity seen in actual tissues. Fast forward to today, and organoids are not just collections of cells. They are dynamic living systems, mirroring the complex interplay of cellular processes that occur within our bodies. Brain organoids, for instance, can now exhibit neural activity, a leap far beyond their initial designs. This newfound complexity allows scientists to study diseases in a more realistic and relevant environment. But it's not just about complexity. Functionality has also leapt ahead. Today's organoids can perform tasks that were once thought to be the exclusive domain of fully developed organs. They can synthesize proteins, generate bioelectrical signals, and even respond to stimuli. This functionality enables researchers to delve deeper into the mysteries of human biology, from disease mechanisms to developmental processes. Let's not forget the advancements in organoid longevity and stability. Early organoids had short lifespans and were prone to structural instability, limiting their usefulness in long-term studies. Today, thanks to advances in cell culture techniques and materials, organoids can be maintained for extended periods, opening up new avenues for chronic disease research and drug testing. The progress made in organoid technology has opened doors to previously unimaginable possibilities. The journey from simple cellular spheroids to complex functional organoids has been nothing short of remarkable. As we continue to push the boundaries of what these miniature marvels can do, who knows where the next leap in organoid technology will take us. Brain organoids today are more sophisticated than ever. The scientific world is buzzing with the possibilities they present. These mini-brains are becoming increasingly complex, capable of mimicking the structure and functionality of our own brains in ways that were once unthinkable. One of the most significant advancements in this field is the ability of brain organoids to model human diseases. By growing organoids that carry specific genetic mutations, scientists can observe how these changes affect brain development and function. This has opened up new avenues for studying neurodegenerative diseases like Alzheimer's and Parkinson's, as well as psychiatric disorders such as schizophrenia and autism. It's like having a working model of the human brain right there in the lab. In addition to disease modeling, brain organoids are also proving invaluable in drug testing. Traditional methods of testing drugs often involve using animal models or cell cultures. However, these methods don't always accurately reflect how a drug will behave in the human brain. That's where brain organoids come in. They provide a more accurate platform for testing new drugs, leading to safer and more effective treatments. But it's not just about disease and drugs. Brain organoids are also helping us understand the fundamentals of brain development. They offer a unique window into how our brains form and function, shedding light on the intricate dance of cells and signals that gives rise to our thoughts, memories, and emotions. This is a pivotal moment in neuroscience. The ability to grow and study brain organoids is transforming our understanding of the brain. It's allowing us to ask questions we couldn't ask before, to probe deeper into the mysteries of the mind. It's important to remember, however, that while brain organoids are powerful tools, they're not perfect. They don't fully replicate the complexity of the human brain, but they're getting closer, and with each new discovery, we're gaining precious insights into the most complex organ in the body. Brain organoids have truly revolutionized the way we approach neuroscience. They've given us a new lens through which to view the brain, opening up a world of possibilities for research and treatment. As we continue to refine this technology, who knows what discoveries lie ahead? The journey of brain organoids is far from over. As we cast our gaze into the future, brain organoid technology stands on the precipice of numerous exciting developments. Imagine a world where diseases like Alzheimer's, Parkinson's, and even mental health disorders are no longer life sentences, but rather conditions we understand and can potentially cure. That's the promise that brain organoids hold. In the near future, we might see the creation of more complex organoids. These might not just mimic the structure of the brain, but could also replicate its intricate functions. This could lead to the development of personalized medicine, where treatments are tailored to the individual, based on their unique brain organoid. Moreover, these advancements could revolutionize our understanding of brain development and neurodegenerative diseases, potentially leading to breakthroughs in treatments and cures. However, with such leaps in technology, we must also anticipate challenges. There are technical hurdles to overcome. For instance, the current organoids lack vascular systems, which limits their size and complexity. Scientists will need to devise ways to incorporate blood vessels or find alternatives to nourish larger organoids. Furthermore, ethical concerns will undoubtedly surface. As these brain organoids become more complex, questions about consciousness and personhood will arise. At what point does an organoid become sentient? And what are the implications of creating sentient tissue in a lab? But let's not forget the potential benefits that outweigh these challenges. Brain organoids could provide invaluable insights into neurological and psychiatric disorders, many of which are poorly understood today. They could also be used in drug testing, reducing the need for animal testing and speeding up the process of drug development. Indeed, the future of brain organoids is a thrilling frontier, filled with potential breakthroughs and new ethical challenges. It's a journey that promises to rewrite our understanding of the brain and potentially revolutionize medicine and neuroscience. The future of brain organoids holds exciting prospects in uncharted territories. With great power comes great responsibility. This saying is especially true when it comes to the field of brain organoid technology. As we delve deeper into the intricacies of this remarkable scientific advancement, we must also grapple with the ethical considerations that accompany it. The creation of brain organoids brings us one step closer to understanding the human brain, offering potential solutions for neurodegenerative diseases, brain injuries, and mental disorders. However, these tiny, self-organized three-dimensional tissue cultures also raise profound questions. One such question revolves around consciousness. As brain organoids become more sophisticated, there is a theoretical possibility that they could develop a level of consciousness. Although this scenario is currently speculative and far from today's scientific reality, it's a consideration that researchers cannot ignore. If consciousness were to occur, what rights would these organoids have? How would we ensure their ethical treatment? Another key ethical consideration is the source of the cells used to create brain organoids. Typically, these cells are derived from human skin or blood cells, which are then reprogrammed into stem cells. This process could potentially involve the manipulation of human embryos, raising ethical dilemmas around the sanctity of life and the boundaries of scientific exploration. Moreover, the potential for misuse of this technology is another concern. Could brain organoids be used in ways that violate privacy or human rights? Could they be exploited for commercial gain or military advantage? These are not just hypothetical questions. They underscore the need for careful regulation and oversight of brain organoid technology. As scientists, we have a responsibility to ensure that our research is conducted ethically and with the highest regard for the potential implications of our work. In a world where science often outpaces regulation, it's crucial that we remain proactive in addressing these ethical considerations. We must foster open dialogue, engage in rigorous ethical analysis, and develop comprehensive guidelines to govern this emerging field. Navigating the ethical landscape of brain organoid technology is as crucial as the scientific advancements themselves. As we continue to unlock the mysteries of the human brain, let's ensure that we do so with both curiosity and conscience. Brain organoids are not just a scientific marvel, but a societal game-changer. Imagine a world where we can test new drugs, therapies, and treatments on brain tissue that closely mimics our own. That's the promise of brain organoid technology. It could revolutionize healthcare as we know it, by providing a more accurate model for studying diseases, predicting drug response, and ultimately, designing personalized treatments. Consider the impact on neurodegenerative conditions like Alzheimer's or Parkinson's. Currently, these diseases are like locked rooms without keys. But brain organoids could provide the keys, or at least a blueprint of the locks. They could help us understand the mechanisms of these diseases at a cellular level, paving the way for effective treatments or even cures. But the ripples of brain organoids extend beyond healthcare. They could also have significant economic implications. For one, they could dramatically reduce the cost and time of drug development. Instead of years spent on animal and human trials, pharmaceutical companies could test new drugs on brain organoids in months. Moreover, brain organoids could fuel new industries. Imagine biotech companies specializing in creating personalized brain organoids, or tech startups developing software to analyze the data generated from these organoids. The possibilities are as vast as the human brain itself. But let's not forget the potential societal challenges. There could be ethical issues around the ownership and usage of brain organoids. There could be debates about the rights of organoids, especially as they become more complex and brain-like. There could be concerns about the misuse of this technology. As with any revolutionary technology, it's important that we navigate these challenges with care and foresight. To sum up, brain organoids have the potential to transform our society. They could revolutionize healthcare, fuel economic growth, and spark ethical debates. But amidst the excitement, we must also tread carefully. We must ensure that this technology is used responsibly, ethically, and for the benefit of all. The impact of brain organoids extends beyond the lab, shaping the future of our society. Brain organoids, a small cluster of cells with an enormous potential. These tiny yet powerful structures have been our main thread of discussion today. We've journeyed from the dawn of brain organoids, highlighting their humble beginnings, to the intricate labyrinth of today's organoid technology. We've seen how this technology has evolved over the years, transforming from a mere scientific hypothesis to a tangible, practical tool. The evolution of organoid technology is a testament to human ingenuity and the relentless pursuit of knowledge. We delved into the current state of brain organoids, discussing the various applications they have in today's world. From disease modeling to drug testing, these cellular structures are proving to be invaluable in the biomedical field. They are not just a window into the brain's intricate workings, but a key that unlocks numerous possibilities in neuroscience and beyond. We then explored the future of brain organoids, painting a vivid picture of what lies ahead. With advancements in technology and a deeper understanding of the brain, the potential applications of organoids are limitless. They could revolutionize our approach to neurological disorders, offer insights into human cognition, and perhaps even challenge our understanding of consciousness itself. However, with great power comes great responsibility. We also touched on the ethical considerations surrounding brain organoids. As we push the boundaries of what's possible, we must also consider the moral and ethical implications of our actions. Finally, we discussed the societal impact of brain organoids. This technology isn't just confined to the labs and research institutions, it has the potential to transform society as we know it. From healthcare to education, the ripple effects of brain organoid technology are far-reaching. Brain organoids are more than just clusters of cells. They are a symbol of our scientific progress, a tool for discovery, and a beacon of hope for the future. As we continue to unlock the secrets of the brain, organoid technology remains at the forefront of this exciting journey. Stay curious, stay informed, and remember, the future of neuroscience is in our hands.